There's something your body does, maybe once every 5 minutes, whether you notice or not. It doubles up. You take one breath, and then, right on top of it, without letting any air out, you take a second little one, a sort of sip, and then you let everything out in a long, slow exhale. Your grandmother called it a sigh. And for most of human history, nobody could tell you why we do it. Now, I'm not a biologist, I'm a physicist. But every so often a piece of biology falls into my lap, and I see something that looks from the outside like a really well-designed piece of engineering. And when I see good engineering, I want to know how it works, not in some fuzzy, poetic way. I want to know the mechanics. I want to know what pushes on what leaks into what switch turns what on. So here is what I want to do today. I want to tell you what is actually happening physically when you do this little trick your body already knows. And by the end, I think you will see that the sigh is not just some soft thing you do when you are sad or relieved. It is a very good solution to a very specific engineering problem. The problem is this: your lungs have a tendency to collapse. Not completely, you understand, not the big airways, but inside your lungs there are these little sacs, tiny ones, about 500 million of them. They are called alveoli. Each one is maybe 1/5 of a millimeter across when it is fully puffed up. If you took all of them and unrolled them into a flat sheet, you would cover something between 70 and 100 square meters. That is more than the floor of a big living room. And all of it is tucked into something the size of a small loaf of bread. Why so much surface area? Because every bit of oxygen and carbon dioxide that goes in and out of your body has to cross the thin walls of those sacs. Lots of surface means lots of traffic. That is a design choice. You want a lot of exchange in a small volume, so you give yourself a lot of little bubbles instead of one big one. Fine, as far as that goes. Except for one thing. When you have a little bubble of water with air inside, the water has a property called surface tension. It pulls inward. It wants to close the bubble up. It wants to make the sac as small as possible. This is why a raindrop is round. This is why you can skate an insect across a pond. The surface of the water is pulling on itself. And under certain conditions, the water film inside one of your alveoli wants to close it up entirely. Snap it shut, like pressing 2 wet plates together. There is a formula for this, worked out by Pierre-Simon Laplace about 200 years ago, and later by Thomas Young. The pressure needed to hold a little sac open against its own surface tension goes up as the sac gets smaller. That sounds backwards. Most people, when they first hear it, they say, wait, what? A smaller bubble needs more pressure? Yes. That is what the math says. And it means the littlest alveoli, the ones doing the most exchange, are the ones most likely to collapse. So your lungs have a problem. Nature has to solve it, and nature does. There is a lipid your body makes called surfactant. It coats the inside of the alveoli. Surfactant is a funny molecule. When the sac is big, the surfactant molecules are spread out and they do not do much. But when the sac gets small, the surfactant molecules crowd together, and they actually push out on the water film, and they reduce its surface tension. The smaller the sac gets, the harder the surfactant pushes back. So the collapse does not happen, or at least most of the time it does not. I want to emphasize this because it is a pretty good trick. You have a material that works harder when it is needed more. That is not common in engineering. We have a hard time designing things like that. Nature does it with a slick. But here is the catch. If you breathe shallowly for too long, and you hold your chest in a little, the way people do when they are worried or tired or hunched at a desk, some of the smaller sacs collapse anyway. The surfactant is good, but not perfect. And once a sac is closed, a regular breath does not open it again. A regular breath is not strong enough. You need something bigger. You need a pressure spike. That is the first thing your sigh is doing. When you take a normal breath, and then right on top of it you take a second small inhale, you push the pressure inside your chest higher than a regular breath would. And that extra push pops open the sacs that had stuck shut. The people who first described this in the laboratory were working in the 1930s. They were studying patients in iron lungs and people in claustrophobic spaces. They noticed something their machines could not do. The patients under stress would spontaneously make a double breath. And after they made it, the blood oxygen measurement went up. The collapse had been undone. We know now that this double breath program is generated by a very small cluster of neurons in your brainstem, and I will come back to those neurons in a moment. But the mechanical part, the popping open of the sacs, that is purely physics. You are doing fluid mechanics on yourself. Every 5 minutes or so, whether you know it or not, you are performing a reopening maneuver on your own lungs. The body is sighing to keep itself inflated. That is already a nice result. If we stopped here, it would be worth the time. But there is more going on. Here is the second thing. While your little sacs are popping open, something separate is happening in your blood. When you breathe shallowly, carbon dioxide builds up. Not by a lot, just a little more than it should. This is because you are not exchanging enough gas. Your blood does not like this. It has chemoreceptors, clusters of cells in your neck, near your carotid arteries, that measure the carbon dioxide level and report it to your brainstem. When the level creeps up, the brainstem says, "Speed up the breathing, we are falling behind." And then something interesting happens in how you feel. Your chest starts to tighten. You start to feel a bit of air hunger. You feel, in fact, like you cannot get a full breath, no matter how hard you try. I want you to pay attention to this next point, because it matters. That feeling of not being able to get a full breath, which a lot of people associate with anxiety, is not a psychological thing. It is a blood chemistry thing. Your arteries are detecting something real. Your brain is responding to a signal from your neck. The long exhale at the end of the sigh is what clears the extra carbon dioxide. You empty your lungs more completely than a regular breath would. The gas balance shifts. The chemoreceptors in your neck quiet down. The signal to your brainstem weakens. The urge to breathe fast weakens. And within 2 or 3 of these sighs, your respiratory rate drops. Not because you decided it should drop, because the chemistry underneath you has shifted. I find this interesting because it tells you something about what we call anxiety. A lot of what we call anxiety is a gas exchange problem that got into the feedback loop. You were worried. The worry made you breathe shallowly. The shallow breathing raised your carbon dioxide. The high carbon dioxide made you feel like you could not breathe. And the feeling of not being able to breathe made you more worried. Around it goes. The sigh is where the loop gets broken, not in your head, in your blood. So far, I have told you about 2 physical mechanisms, surface tension and gas exchange. Both purely mechanical, nothing mysterious. The third thing is about nerves, and here I have to be careful, because the nerve part of the story has only been figured out in the last 10 years. Before that, we knew that slow breathing made people calmer. Yoga teachers knew it. Every grandmother in every country knew it. But nobody could tell you which neurons did it, or how. The nerve I want to tell you about is called the vagus. It is the 10th cranial nerve. The word vagus is Latin for wanderer, because this nerve wanders. It leaves the base of your skull, goes down through your neck, fans out into your heart, your lungs, your stomach, your intestines. It is the main output line of what doctors call the parasympathetic nervous system, the system that tells your body to rest, to digest, to slow down. Along the airways of your lungs, there are little stretch sensors. When you take a deep breath and hold the air for a moment, these sensors fire. They send a signal up the vagus nerve back to the brainstem. The brainstem reads that signal as, "We are relaxed. We have time. The crisis is over." And in response, the brainstem tells your heart to slow down and shifts the whole body toward a calmer mode. Now here is the part that a lot of people miss. The exhale is more powerful than the inhale for doing this. When you exhale slowly, the stretch sensors in your lungs unload gradually, and they send a long, sustained train of signals up the vagus. The brainstem gets more information. It has more time to shift the settings. A fast, choppy breathing pattern sends short, clipped signals. It does not have the same effect. The nervous system reads it as, there is an emergency, keep everything at full alert. So the reason the long, slow exhale matters physiologically is that it gives the vagus nerve time to do its work. In 2023, a group at Stanford ran a randomized trial. Melis Yilmaz Balban was the lead, along with Andrew Huberman and David Spiegel. 111 people. They compared this psi-based breathing against 3 other things. Against a pattern called box breathing, where inhale and exhale are equal. Against another pattern where the inhale was longer than the exhale. And against simple mindfulness meditation. 5 minutes a day, for a month. The people who did the psi pattern improved the most on the measures that matter. Lower resting heart rate, lower anxiety scores, better mood. The paper came out in a journal called Cell Reports Medicine. I encourage you to look at it yourself. Let me be honest about the limits. One study is one study. The mechanism is solid. The replications are still coming in. I would bet on it. But I will not oversell it. Now I come to the last part, and this is the part that surprises me the most. In 2017, a group at Stanford, working with Jack Feldman's laboratory at UCLA, published a paper in the journal Science. Kevin Yackel was the first author. Mark Krasnow was the senior author. Feldman at UCLA had found, back in 1991, a cluster of about 3,000 neurons in the brainstem that generate the rhythm of breathing. He called it the pre-Butzinger complex. It is the breathing pacemaker. 3,000 cells that decide when you inhale and when you exhale. Inside those 3,000 cells, Yackel and Krasnow found a smaller group, about 175. They identified it by the specific genes those cells express. And they did something bold. In mice, they destroyed just those 175 cells, and they left the other 2,800 alone. The mice still breathed normally. The rhythm was fine. But something else changed. The mice became calmer. They spent less time in aroused states. Then the team traced the axons of those 175 cells. And they found that the axons went directly to another region of the brainstem called the locus coeruleus. The locus coeruleus is small, a little blue pigmented spot, about the size of a grain of rice. It contains somewhere between 22,000 and 51,000 neurons. That is it. And those neurons are almost the entire source of a chemical called norepinephrine, which is what your brain uses to make you alert, vigilant, anxious, on edge. So what Yackel and Krasnow discovered is this. 175 neurons in your breathing center talk directly to the anxiety center. When breathing is fast and shallow, they fire hard, and the anxiety center fires hard. When breathing is slow and deep, they fire less, and the anxiety center quiets. They are a pipe, a direct line from your lungs to your mood. This is the thing that pharmaceutical companies have been trying to do chemically for about 70 years. Benzodiazepines, the drug class that includes Valium and Xanax and Ativan, work by quieting the locus coeruleus. They bind to a receptor on those same norepinephrine cells, and they make them less active. That is the final mechanism. Same endpoint, different route in. So when you do a slow, deep sigh, you are, quite literally, turning the same dial a pill turns. The dose is smaller. The effect is shorter, but the dial is the same dial. Now I want to say something about sleep, because there is an honest comparison here that I do not want to skip. Sleep also quiets the locus coeruleus. During deep, non-rapid eye movement sleep, it fires at about 1/3 of its waking rate. During rapid eye movement sleep, it fires almost not at all. This is part of what sleep is for. long stretches of quiet for the anxiety center. But there is a practical problem. Sleep takes 10 or 20 minutes to start. And during those 10 or 20 minutes, the anxiety center is still running, which is often what is keeping you awake in the first place. The sigh does not replace sleep. It cannot. But the sigh begins to do its work in seconds. When the crisis is faster than sleep, the sigh is the right tool. Let me try to pull all of this together. You have a body that contains in its chest 500 million little bubbles with a surface tension problem. You have a surfactant slick that fights the tension, but not perfectly. You have a small program in your brainstem that, every few minutes, pops the stuck sacs open with a double breath and clears out some excess carbon dioxide with a long exhale. The popping and the clearing together send a signal up a nerve that wanders through your whole body. That signal tells your heart to slow and tells a small group of 175 cells in your brainstem to quiet down. Those cells in turn tell the anxiety center of your brain to ease off. All of this happens when you sigh. And all of this, as near as I can tell, has been going on in your ancestors for tens of millions of years. Birds do it. Mammals do it. We inherited the machinery. What I find most satisfying about this story is that it is an honest piece of physics doing an honest piece of biology. Nothing magical, nothing mystical. Pressure, surface tension, gas diffusion, nerve impulses, synapses. The same kinds of things we study in laboratories, just arranged inside of you in a way that quietly keeps you alive. Before I go, I want you to try it. One time. Take a breath in through your nose until your chest is full. Take a second shorter breath on top of it. And then let it all out, slowly, through your mouth until your lungs are empty. You just performed a small act of fluid mechanics, gas exchange, nerve signaling, and brainstem circuitry. So I will leave you with a question. When you did that sigh, just now, what did the next 10 seconds actually feel like in your body? Tell me. I am curious what you noticed.